Hi, I'm Natalie Leger. Let's talk about biotechnology and the genetic modification of prokaryotes. So, microbiology provides the playing field necessary for genetic modification. Biotechnology currently relies heavily on genetic modification for pharmaceutical, agricultural, medical, and food safety advancements. By creating genetically modified organisms using rDNA or recombinant DNA technologies, we can create hardier crops and livestock, create bacteria that makes our medicine, and produce life-saving vaccines. So here's an example of how we genetically modify a bacterium. So up here we have Vibrio fischeri, a marine bacterium that bioluminesces. So it does that using the Lux operon in its DNA. So this Lux operon is going to code for luciferase, an enzyme that allows it to bioluminesce. So what if we want a bacteria that doesn't normally do this to also bioluminesce, just like Vibrio fischeri? So we would start by cutting it out using restriction enzymes, and we would cut out the specific part of the DNA, the Lux operon, and put it into a plasmid, making this operon and genes mobile. So this plasmid is going to act as a cloning vector, and it's going to enter our target bacteria, E. coli for this example. And once the plasmid is inside the bacteria, or the bacterium, it's going to create recombinant DNA and combine with the chromosome, the bacterial chromosome that's already present. So once this happens and this E. coli multiplies and clones itself and grows into a colony, that colony is going to bioluminesce just like Vibrio fischeri using the Lux operon that's now part of its chromosome. So some of the ways that this genetic modification for bioluminescence is helping science right now is it provides an alternative way to easily measure bacterial growth. And there's experiments where it can detect pathogenic bacteria in food, showing us where the bacteria is because it's bioluminescing. It can act as a detector for the efficiency of RNA polymerase. It can also act as a detector for the efficiency of antibiotics in laboratory animals because if the pathogenic bacteria is genetically modified to bioluminesce, as the antibiotics kill it off, the bioluminescence will decrease in the laboratory animal. Thank you for watching. Here are my references.